Hi, everybody. My name is Denny Lee, and welcome to our first data and AI online meetup right after Labor Day. So today's session is the genomic variant data ingest and manipulation using GLOW. GLOW is an open source toolkit for population genetics, and the project is an industry collaboration between Databricks and the Regeneron Genetics Center. In today's talk, we'll focus on how to use GLOW biobank-scale genomic variant and annotation can be easily ingested from legacy flat file formats into Spark DataFrames and stored as delta tables. We will then show that GLOW provides multiple functions and transformers to easily perform variant QC and data manipulation at scale. We are going to actually have a pretty full session today, so please ask all of your questions in the Q&A panel, and we'll do our best to answer them live in the panel. If you're actually on YouTube live, ask your questions in the live chat, and again, we'll do our best to answer them. So my name is Denny Lee, and I'll be the host for today's Data and AI online meetup. I'm a developer advocate at Databricks, working with Apache Spark since 0.7. Prior to Databricks, I was at Microsoft and SAP Concur, and I also happen to have a background in healthcare life sciences with a master's in biomedical informatics. I'm also really happy to have Amir and Gavesh presenting today's session. I'll let them introduce themselves. So let's start with Amir, as he's going to be presenting the first section of the session.
Thank you so much, Danny. Hi, everyone. This is Amir Kermani. I'm a solution architect here at Databricks. I've been here for 2 years now to date. Prior to Databricks, actually, I worked as a senior data scientist in big data and e-commerce space, but my real background was, prior to that, I was one of the first staff scientists at Ancestry DNA when we were just, starting. We were around 1 million samples then. And my training actually is in mathematical biology and statistical genetics. I was a postdoc before that at Howard Hughes Medical Institute, focusing on population genetics. I'm really happy to be here.
Hi, everyone. I'm Kiavesh Kiamfar. I'm a senior software engineer at Databricks, working in the health and life sciences team and focusing on the genomics runtime at Databricks and Project GLOW. So along with my colleagues in the engineering team, we have developed the Project GLOW in collaboration with the Regeneron Center. Before joining Databricks, I was an associate professor at Texas A&M University, where my teaching and research was focused on algorithm development and optimization related to bioinformatics. And I've joined Databricks to continue that path in the industrial setting. Thank you to be with you. And I guess back to Amir to start the presentation.
Thank you, Kiavesh. Okay, so let me just start sharing my screen. That's the fun part of every starting presentation. Okay, all right, so what we are going to cover today is, in the beginning, I will start just a refresher for those who are not familiar with Project GLOW, just to, just provide some background about where it comes from, what is our philosophy in developing, yet another bioinformatics and computational biology tool. And I hope, at the end of it, you have an appreciation why this is actually a very different approach in doing genomics and working with genomics data. And then, for a few minutes, I will do a live demo of it, and then I hand it over to Kiavesh, who is actually the person who has really heavily been involved in developing the actual library and the tools, and he will go really have more of a deep dive here. Okay, so, first thing first, you might have seen this graph, why we are talking about genomics at scale and why scale is an issue now. The number one thing, just looking at the history of basically the cost of sequencing from this first draft of human genomes to, the current day, we see a dramatic drop in the cost. It's beating Moore's law, and it is now cost-wise, it's hovering around, $1,000 around per genome. As a result, and you have a lot of initiatives to sequence human, every individual to be sequenced, because there's a lot of health-related data, obviously, and we'll talk about it, like what's the value in this. But from a purely technical perspective, when you look at the size of the data, it's pretty fascinating that when you look, for example, just the ballpark is we think that by 2025, the size of the genomics data would be the sequence and that year would be 25 times more than all the videos uploaded to YouTube. This just signals purely from a technical perspective that we have a scale and a big data problem. Also, you're seeing a lot of initiatives around the world of the biobank data sets that are coming out. Famously, UK Biobank, it's more than 500,000 whole exome sequences, along with a lot of phenotypic data, more than 20,000 phenotypes straight from EHR, EMRs. Regeneron recently, like a couple of months ago, surpassed 1 million samples. You have the FinGen project. Also, on the commercial side, my old company, Ancestry DNA, I haven't really followed up, but the last time I looked, you have more than 16 million samples. Similarly, 23andMe, it's more than 10 million. And all of these, although they are array, they are also capabilities to switch to whole genome sequencing, as Ancestry DNA actually is doing it with Ancestry Health. And, why do we have all this, all of these data sets? Why do we have all these initiatives? Well, obviously, you see our FinGen one, the heavy hitters of genomics that is like now heading that effort and this whole, initiative to build this amazing, research centers. And this is something actually I copied from, the Twitter, Adam from 23andMe just, they just recently published, a really interesting study on, association, just looking at, genetics and non-genetics causes of, factors influencing COVID. All of these are possible because when you're looking, from complex diseases in particular, you need a lot of data sets to get any, any significant, for example, when you're looking at association studies, you need a lot of samples to get any significant results. And, this is why, and what we are anticipating that this to grow, that you're going to have a lot more data sets such as this one. So, and as I mentioned, this is just the beginning. So we have a problem with scale, but when you're looking at scale, scale can be viewed from two ways. Solving scale problem is that one of the things is that just you throw machines at it, right? So you have big data, you can, just better hardware, faster algorithms, and distribution. And this is what we'll talk about. Like, this is what Spark is shining as, just distributing, abstracting away all of these complexities of dealing with algorithms from the user. The other side of scale is when it comes to the people side. So you have more and more, I mentioned, for example, UK Biobank. You have the genomic side. You have a lot of people who are working on the genomic side, sequencing genomics data that we have specialized tools for it. Historically developed within the compile bioinformatics community. But also you have data sets, for example, I mentioned electronic medical records. And all of these, you do a lot of like natural language processing workloads. You have even, scraping image data, extracting features from images, whether it is, medical, images, or like, basically getting text recognition from like the doctor's handwritten in a note. This is, you can extract a lot of phenotype data, but that falls within what traditionally we call data science or ML workloads. So in order to facilitate collaboration between teams, we need a platform that addresses both the technical challenge of scale, but also on the people side, that people can be working together and having the same tools to work together. And this is actually, regardless of CompBio, I think the history of Spark, when you look at it, is coming as a result, as a response to this. Like, this is a unified analytics engine designed for big data processing. And it supports, streaming, SQL, machine learning, graph processing, and originally actually it starts from within a UC Berkeley Amp Lab. And I like, when you say Amp Lab, it's like, algorithms, machines, and people. So that is what the idea is, that also we are looking at scale and just having algorithms for people. So, in 2009, the team developed Spark, but then they later donated that to the Apache Foundation. And it is an open source, project. It's I was just looking actually at some reports, like the second only to React in terms of popularity among all of these other open source, solutions out there. So huge community around Spark, predominantly because it's performance, and also it's just following, support for multiple languages. And you can do end-to-end work within the Spark data, framework. As I mentioned, it supports Scala, Java, Python, R, and SQL. So, different personas can work in the same space. So, collaboration is, increased. And also, we have, in terms of contributors to the project, we have more than 1,500 developers. And I'm sure Danny has more up-to-date stats on this. And it is like more than 200 companies are contributing. So we identified this is a really good ecosystem that it is basically the de facto, it is a de facto platform for doing big data and machine learning. So later what we are getting is, so now that we are looking into the bioinformatics, space, as I mentioned, we have a big data issue. So we have problem of scale. What is the best tool out there for addressing scale, both from being an adapted ecosystem that people are working with, and also being something that can address machines and scalability? We look at Spark. So GLOW project that we actually announced, it was last year, ASHG, it's developed in partnership with the Regeneron Genetics Center. It is built on top of Spark, but also it supports native Spark APIs, so that you don't actually have to do, this basically where it goes on the Spark, built on the Spark DataFrames. So the same way that you're going to query any table, as you will see in the ingest part that I'm talking about, the same way that you're going to query any data set, you're going to query genomics data sets. And also, because this is within this DataFrame API, it's really easy to merge genomics and phenotypic data. You can use Spark to scrape, to do feature extraction from your EHR, EMR, or the image data, and then merge that with the genomics data set. Also, it is, as Kiawesh will go through, we have also considered this, that you are going to have, for example, use cases that you want to bring your own functionality and just use the distributive power of Spark for that. So that is what we are going to talk about. So in terms of from ingest side of the work with GLOW, so suppose that we are starting from a VCF file, traditional, a genomics format, or BGEN or PLINK. These are like, your data sets arrive in flat files in that form. Now you want to ingest it into a Spark DataFrame. So this can be easily done just by, similarly, the same way that you would say Spark read format, let's say CSV or XML, you can just write VCF, BGEN, or PLINK, and specify the path where your data is. So keep in mind that we have codecs. You don't have to specify anything in terms of the encoding. It can be from gzip, bgzip, or just flat VCFs. Now that you ingest the data, the next thing is that we are assigning a schema to it. So familiarity with schema, basically in all data, anything with data, you have a table, let's say, and it has some columns. You can think about it, and then what are the types of those columns? Some of them are strings, some of them are arrays, etc. When you're ingesting the VCF file, basically the table or the data frame that you get every row corresponds to a variant, so be it indels or SNPs. And then you have all the information, all the attributes of that variant, physical location, or start and end, or like alternative allele or reference allele, and all the info fields that you see in a BCF file. Actually, it is ingested, given structure, and it appears as separate columns. Also corresponding to the genotypes, you have the genotypes column, that it's an array of, you can think of, if you're not familiar with struct, you can think of like dictionaries in Python. Let's say for 1,000 genome project, when you're ingesting it, that genotypes, every row has an array of length 2,500. Each element corresponds to a sample, which includes sample ID and the actually called genotypes. And if it is phased, obviously you have order matters. This is an example of ingesting, for example, the data. So you have chromosome name or contig name, also start and end, RSIDs, and you can go further, look at the genotypes values, and as I mentioned, you have sample ID and calls. However, you have a lot of options. You can, for example, exclude sample IDs for if you don't need that directly because the order is preserved. And other options that you can look at the docs, and Kiavesh will talk more in detail. The other part is, now that you have ingested the data, you want to do some analysis on it. So the way we are looking at the analysis of genomics data is like any other data analysis. You basically have an aggregate, you have a grouping of some data points, and then you want to run an aggregation. So this grouping can be, in this case, for example, on the grain, we have some basic summary statistics functionality involved. Included in GLOW, the ones that are going per variant. So in this case, call summary stats. Basically, you call that on the genotype column, and what you get as an output is basically for every row, you will get the allele frequencies, the homozygosity, number of heterozygotes, etc. And similarly, you can repeat that for other functions. For example, you want to calculate Hardy-Weinberg disequilibrium values for a pre or GWAS. You want to exclude some of those that are outside, they are not in Hardy-Weinberg equilibrium. Then you can just apply that function. And this is something that we are actually adding constantly to this functionality. But the whole idea is that you call statistical genetics process, routine, the same way that you would call any aggregate function in Spark SQL. Similarly, if you have sample level summary stats, for example, you want to look at homozygosity, homozygosity or heterozygosity of individuals, that you can, for each individual, let's say you want to calculate down the road ingredient coefficient or anything. This is really done within this sample level summary statistics, and then later you can do your own analysis the same way. By the way, I want to keep in mind, if you are also familiar with pandas for data engineering, you actually you don't want, you do some initial analysis using PySpark APIs, but then you can convert your data set into a pandas-like data frame, which is a koalas project, called koalas API, which the whole idea is that you can still run pandas code, but under the hood, it's going to run Spark for scale. Okay, so now that you have done all this analysis, the next thing that you want to do is you want to write it. So you did some analysis. Let's say you have ingested 1,000 genomes data, ingested UK biobank. You did some QC, some filtering, a subset of the data. You want to write it back as, let's say, a VCF file. So you ingested the BGEN, you want to write it a VCF, and this is a smaller file that you can just do it in one file. You can use the big VCF format that will output the traditional VCF flat file. Alternatively, you can actually shard this file if it's a bigger file and you want to have the same way that you have like sharded like Parquet file or CSV that we distribute. You can do that with just writing VCF. However, one of the big advantages of Glow, in my opinion, is its native integration within Spark ecosystem. So if you ingested the data set in a data frame, Spark data frame, now you can easily write it into the Delta Lake. And Delta Lake is something that Kiawesh will talk about more, but the whole idea is that this is the next generation engine on Spark that brings it brings a lot of performance and what more importantly, reliability of data warehousing in terms of asset transactions and all of that good stuff into a data lake, which is something that scientists are most often, all our data is in a data lake, but we don't have much of a governance around it. For example, we don't have data versioning, and it's not easy to do that. But if you write your data in a Delta, it comes with it, like, for example, you have data versioning. You can go back to query, for example, from a specific snapshot of your data set. So you just don't worry about it. You just constantly, you get updates from UK Biobank, you write in the same data lake, but then you can go from any specific freeze of the data, you can do your analysis. And this is super important in terms of reproducibility of results, because you should know, now it's very easy to know what data were used, let's say for this GWAS that we had. Finally, I encourage you to check this. We have a Slack channel for Glow project. Also, we have a Google groups, and this is the GitHub page. But just quickly, I talked about all of this, what Glow is about. Maybe quickly going through a quick, live demo. It's when you have live demos, there's always a risk, but I take my chances. By the way, I'm running this on the community edition of Databricks. Keep in mind, you don't have to run Glow on Databricks. So it's open source, and the documentation actually is geared toward you have your own installation. But for ease of use, I'm using Databricks, which is, but also this community edition is accessible to you, it's free. So you can just sign up and start, playing around. So the first thing I do, I'm running Glow on a Genomics runtime. The first thing I do always, this is by the way, this is a Python library, so I'm importing I'm just doing the import on getting the Glow installed. As you see in my cluster that I'm running, this is the Genomics runtime here. It just gives you the latest, I'm running the latest runtime on Databricks. Now we have on a beta is actually we're at 7.3. So within every Databricks deployment, actually, you have a Databricks data set that is included. So we have actually VCF files from 1,000 genomes. These are just VCF files from chromosome 22. And this is just, to play around and look at the data for you. So what you're seeing here is I can just give it a path to, let's say, this first file, which is a VCF file in GZ format. So I give it a path. And then next thing is that I'm going to use, maybe I better have it magnified. I'm going to use this data set and ingest it into a Spark data frame. So I just use Spark read format, I do a VCF, and then load the path. So now the path is loaded. You see, it is giving the schema that I wanted. So every row corresponds to a cantique, chromosome, start, end, and all of that. And if you want to visualize it, I just, within Databricks, I use DF display. Outside of Databricks, for any Spark, you can just say df.show, and it shows you snapshot of your data. As you see, we have all of these, info fields, for example, are expanded. Every column is one info field, for example, African allele frequencies or other data sets that other values of interest that you want to see. So next, now that I have this, next, let's say I want to do some basic analysis. As I mentioned, you can just call this, for example, select expr is a Spark SQL command. So I'm going to select some of the columns, let's say for simplicity, just I'm gonna take start position. And also now I want to calculate the allele frequencies. To do that, I'm going to use a utility function within Glow, which is expands struct. That basically opens up a struct, and every key now appears as a column. And then I use call summary stats on the genotypes column. Now this is done. Now I can take a look at this. By the way, if you're new to Spark, sometimes you see that, for example, this comment took, sub-second, very, very fast. But then you say, wow, this is really fast. It's not actually, what you're seeing is that it's just like, it lays out the execution plan. And later when you call an action on this data frame, actually that is being executed. So now I say display. Now this is where actually the execution of this is done and the Spark jobs are ran. So now you see that, for example, I have, start and end, and all these values, for example, the number of heterozygotes, et cetera, that I want. So from this, now I can just go ahead and just write a query. Let's say I want to look at, select from this. Actually, filter is an easier way of doing it. All those that the number of heterozygotes is less than 10, for example, right? So this is just a filter command. So this is to give you an idea how actually your analysis, you can incorporate genomics, and you can incorporate your traditional genomics workloads and any other thing. The other last thing I want to talk about, I mentioned, the same way that you are looking at genomics data sets. Let's say I have I want to get some phenotype data, right? Next, I have that. Okay, maybe I am not looking at this. Let me just do it again here. That should be right. Okay. Okay, there you go. So within the same Databricks dataset, these are simulated data of electronic health records. Let's say I want to get conditions of patients, and hypothetically imagine that these are the same patients that I have a sequence for. I can just do the same thing, spark.read, but this time format, I can say it is CSV, although for your information, for Spark, actually specifically, we have this different API that you can directly say CSV. But to give you an idea, it's the same as writing a VCF. For example, header is true, and then load the path. So now this is what I have is that I have all the conditions, right? So if I want to do combining genotype and phenotype data, assuming that this was the same data set, I would just, for example, if this sample size, samples were the same IDs at 1,000, the sample ID that I have for the genomes, I would just simply say dfehr dot join or merge actually with this other one, join on this one on whatever column that you have. So basically, it's a SQL join. Okay, so if you're interested, actually, you can. I have if you want to download this notebook that we generated, you can just download and import it within your own community edition and start playing with it. This will be the link that you can use to get the data. On that note, I'm going to pass this to Kiarash. Thank you very much.
Thank you, Amir. I think, Amir, you have to unshare your screen.
Yes, sorry about that.
Okay, great. Awesome. So I'm gonna pick up where Amir left and talk a little bit more about genomic variant annotation ingest into Spark DataFrame using GLOW, and then follow up with some overview of the genomic variant manipulation capabilities that we have designed into GLOW. If you're familiar with bioinformatics, I think many of the capabilities that we have incorporated here will be of interest to you, because we have, as Amir mentioned, our motivation here is to bring virtually unlimited scalability to these operations, where they can be done easily on biobank-scale variant data sets and annotation data sets. Just to complete our data ingest perspective, Amir talked about ingesting variant data into from VCF, BGEN, or PLINK format into a Spark DataFrames. And I want to mention that in GLOW, we also have a genomic annotation data ingest from GFF3 format. If you have worked with genomic annotations, you know that this GFF3 format is one of the very common formats to store genomic annotations. And we have a data source in GLOW, which also can be similarly used to ingest the annotation data related to different parts of a genomic sequence into a Spark DataFrame. So just to emphasize here, again, our philosophy in GLOW is that we kind of support all data ingest from the traditional flat file formats like VCF, BGEN, and PLINK, and get the information out of these formats and put them into the data engineering, the modern data engineering and database structures that data scientists and data engineers use, so that they can be easily queried and manipulated. So when you ingest this data into a Spark DataFrame, then you can easily work with it as any other database that you work with using Spark commands. And at the same time, you can store your data frames in Delta tables. Delta is a project open source by Databricks, where you can refer to it at this link and learn more about it. But essentially gives a lot of optimizations and performance and new features to the Spark ecosystem for the storage and retrieval and querying of the data, including ACID transactions and versioning of the data. So you can have different versions of your variant or annotation data over time and go back and forth over them. And this can be done very seamlessly with very efficient APIs. Today, I just want to do a little bit of a more deep dive into the features that GLOW has in regard to the ingest of the annotation data and the later variant manipulation. So annotation data can be ingested into Spark DataFrames, and the details of that is given on a blog post that is linked here, and you can find it on GLOW documentation website if you go to projectglow.io. But if I have the variants and I have the annotation, the job of annotating your variants will be just as easy as doing a join between two database tables that you have, and these details are explained in this blog. I just want to talk a little bit about the ingest of annotations before moving on to the manipulations. So GFF3 format, if you are familiar with it, is a simple tab delimited text file with 9 columns, where the first 8 columns are you can read about their definitions in the website of the GFF3, but the last column attributes includes the gist of annotations. So for a particular sequence, it talks about different properties that sequence has, like ID, like the parents of that sequence, and many different things that can you can annotate, gene ID, transcript ID, its exons, and so on and so forth. So we have designed this GFF reader to be quite versatile. So essentially what it does is that it loads this as a Spark DataFrame, which will have a schema as follows, that the first 9 columns will appear with the particular files in your data, for particular types in your data frame. But the last column, instead of just taking this text string, which is semicolon separated, into just a single column, we parse this and generate 1 column for each one of these tags that are in the last column. And this way we create a table which for you has all these tags as separate columns, and every variant that has that particular column will be populated accordingly. Some of these tags have been defined in a standard of GFF3 to be the so-called official fields, and we retrieve them with the very proper types, and even if they are array formats, we retrieve them as array. And then you can have your own files as well, your own fields as well, which we just parse them as string columns and add to the data frame. So I can quickly show this to you as well in a demo notebook. So I don't want to go into the details, but here I have defined a particular path to a particular GFF. This is the genomic annotation downloaded from the RefSeq website for human genome. And just loading it will be as easy as saying a Spark read format GFF and load the path. And if you just load that, you can see that you can look at the schema of this file with this command print schema, and you can see that the base fields are here. Any of the official fields that are parsed and exist in the file are here, and then there are many other fields specific to this particular GFF that are also parsed with the string type, which are added here. And if you look at this data frame, you can see that all these fields that have been parsed. And what is good about this, the good thing is you can easily query this with any of the Spark APIs that you like on any of these columns at a scale, and you have it right in hand. You can join this data frame with your variant data frame and create an annotation of your variants as well. So back to our presentation here. The GLOW, in addition to data ingest, offers several APIs to manipulate variant data. So Amir talked a little bit about the variant quality control APIs that we had. We also have sample quality control APIs. We support coordinate and variant liftover with just a single transformer or functions. We can easily do variant normalization at the scale using GLOW, or do the splitting of multi-allelic variants, which is normally done as a precursor to many downstream genomic analysis. And we even have a very cool feature that if you have a particular custom bioinformatics tools that is a command line tool, we can support parallelizing it using GLOW with something we call a pipe transformer, which I will talk about briefly. So one thing I want to mention that most of the APIs in GLOW are either of our two formats. They are either designed as Spark SQL functions, and they can be used as functions, as functions that are applied on columns of a Spark data frame. So you get your particular columns and give it as an input to your functions, or they are transformers, which you can apply. The concept of a transformer is that you provide a transformer name and the input data frame and some options, and what you get is an output data frame that this transformation has been that is the result of applying this transformation on the input data frame. So please have these two formats in mind as we talk about some of these features in the rest of this presentation. So let's talk a little about the quality control APIs in GLOW. These were covered by Amir, so I'm not going to spend much time on this, but these are of the type that are SQL functions. So if you have, for example, a VCF, ingest a VCF, you get a data frame with these columns using GLOW. There are many other fields as well, probably your info fields in a VCF, but each one of these functions gets some statistics on each variant, and they are usually applied on the genotypes column, which has the bulk of the information in the VCF, which includes the sample IDs and calls and the allele depths for each sample in your variants. So these functions are applied on all the samples for a particular variant, and that's why we call them variant quality control. Now, we similarly have sample quality control functions, which include sample call summary stats, sample depth summary stats, and sample genotype quality summary stats, which you can read more about in the documentation. But here I have just summarized the inputs to this. You can see that this one takes 3 columns of your data frame, which would be genotypes, reference allele, and alternate alleles as the input, and then produces an output. Output is going to be another column, which is output is going to be another column, which is an struct column that includes all of these statistics. Sample quality control is applied across all variants for a single sample. So these functions work on sample 1, for example, across all variants and give you a statistics for them. And then the same thing on sample 2, sample 3, sample 4, and so on and so forth. There are example notebooks for this on the GLOW documentation website, which you can refer to, and they are similar to what Amir showed you a few minutes ago. Now, Liftover is another operations that usually bioinformaticians do before starting working on the data, if your data is not on the right genome assembly. So Liftover, as you know, is the process of getting your coordinates from genomic coordinates from one reference assembly to another. For example, for human genome from assembly 37 to 38. So we have a parallelized liftover functionality embedded in GLOW, which with a single line of code you can do this. Actually, we have both the function and a transformer for this. So in the function form, you provide, you have a Spark DataFrame. The name of the function is liftover coordinates, and this is based on the UCSF liftover tool that is the standard usually used. UCSC liftover tool that is the standard usually used for liftover, and the output of this will be a structure of the lifted coordinates, which is the contig name and a start and end. We also have a liftover variance transformer, which is based on the Picard liftover VCF tool, if you have worked with this. But what's the difference? The difference is that it's scalable. The algorithm is based on this, but you can apply it on gigabytes and terabytes of variant data. So how it's used is the transform paradigm. Liftover variance is the name of the transformer you call. You give it an input data frame and some options. Options are here are a chain file and a reference file. Chain file is, if you have done liftover, you're familiar with it, you need a chain file from a particular genome to another genome, and then you have a reference file, which is the target reference genome that you have. So you have to provide the path to this file and the path to this file. This one does a little bit more than just a simple liftover. That is, it does reverse complementing and left aligning of variants, or the reference alternate alias swapping, just like the Picard tool does. The cool thing about this is that it also gives another extra column, it generates an extra column in your data frame that shows the status of your liftover operation. Variant normalization is another the thing that we support. This is also implemented as a function and as a transformer. Variant normalization, if you're familiar with it, just makes sure that your same variants are represented identically in your data set. And that means that you any variant can have different representations, but by definition you want to have your variants at a particular representation, which you want them to be parsimonious and left aligned, meaning that they have to be to the leftmost possible location in the genome, the presentation. So you can read about this more in the blog that we have regarding this on GLOW website. The way this is done is again using a function or a transformer. Here I talked about the transformer for the sake of time. So again, you have normalized variant input data frame. You have to provide the reference genome path, and there are options to talk about whether you want to replace the current columns with the normalized one, or add them as a new column. I can show you an example of this on a notebook that we have here. We import GLOW, register it, and the reference genome and the VCF path that we have. We load the VCF, as you can see here. Just have a look at the data frame that is generated, and then we call this transformer, which essentially GLOW.transform, name of the transformer, normalized variant, and original variant data frame is the input, and the reference genome path is the reference genome path that we defined, which is the 38 genome path, because this VCF is in 38 assembly. And as a result of this, you can see that the columns are from here are normalized and you have the new columns and coordinates here, and the reference allele and alternate allele are also replaced accordingly. You can have the option without column replacement, which means that the original columns are kept, but at the same time, an additional column is added, which we called normalization results, that shows the normalized things, normalized coordinates as well, and alleles, and as well as whether the normalization was successful or not. Back to presentation. We have a splitting of multi-allelic variants. This is a rather complex operation as well, and we have implemented the best of the breed algorithm that is available for this, which comes out of the variant tools package, decompose tool, and I don't want to go into the details of this, but essentially, if you have a multi-allelic variant, this transformer splits it into biallelic variants. The use of the transformer is very similar. Again, the name of the transformer and the input to the input data frame. What this does is that it splits all the multi-allelic attributes, info attributes, as well as what you have in your form, in your genotypes fields, properly between the two alleles. And this process happens both on arrays, like here 3 and 2 is a split between the two variants accordingly in a correct format, as well as the attributes like this in the genotype field, which are the probabilities which come in the collects order. They are split correctly between the two. So this complex operation is done, and you can do it at a scale. And I will wrap up the presentation by talking about the cool pipe transformer that we have in GLOW. So what is a pipe transformer? So let's say, some of the functionalities we have designed in GLOW are, they have their own APIs, but let's say you bring your own bioinformatics command tools, like let's say BET tools, and you want to scale it to the, so that it can be run in a parallelized fashion on a cluster. So you can easily do that with our pipe transformer. What our pipe transformer does is that it gets, just again, it's a transformer. So the name is pipe, and it gets an input data frame. But what it does is that you have, after you have your data frame, which is partitioned over a cluster, it can be a very large representative of a very large data set. The pipe transformer gets that, and based on an input formatter, converts the data frame into the format that your command line tool understands. Let's say you have a BET tools and you want to use one of the BET tools command that gets as an input a VCF and gives you as an output a VCF to you. So this pipe transformer gets your data frame, converts it to a VCF, pipes it through your BET tools, gets the output VCF, and then based on an output formatter, converts the output VCF back to the data frame that you have. And this way you will, and this happens in parallel, every partition of your data frame, this is the same thing happens on. So as a result, you can essentially apply BET tools at very large scale instead of on a single node. So if an operation that may take several hours using BET tools, or even may never finish because of memory issues, can be easily done over a cluster. So input formatters and output formatters that we currently support are VCF, CSV, and text input. So if your tool can understand VCF or comma separated or tab separated format or text and as input and output, you are good go with this tool. Let me just quickly show a demo of this as well. So the pipe transformer, to be able to run the pipe transformer in your Spark cluster, you have to make sure that the tool that the command line tool that you are using is installed on every node of the cluster. So here I'm going to use BetTools intersect command to find the intersection of my variants with a particular bed file. Therefore, BetTools should be installed on every node. That I have done in this cluster that I have running here before. So I import the required glow and other functions that I may use. And here you can see that I load a VCF of the chromosome 22 of the 1000 genome project. And for the demonstration purposes, I've just limited it to 1000 rows of this VCF. So just looking at the 3 columns of this VCF, we can see them that everything is on chromosome 22 and start and ends are here. Now, to use the the intersection with bed file, I created just a synthetic bed file here. So this is a bed file with 3 rows, which and 3 columns, which chromosome 22, and some ranges start and finish that I want to find the intersect with, and I've saved it in this path using this command. So what I will do is that I have to create the command that I want the pipe transformer to use. That command will be given as essentially a JSON formatted array. So for that command, I've written a string here. So essentially, I would like, this is the command I want to run on my file if I was in command line. I want to find the intersect of the input. So BetTools is my tool, intersect of the input to the the standard input to the output that is my bed file is specified by this, and the output is going to be from the original rows of the input file. So this will be saved as a command that is passed to the bash. So essentially my command will be bash -c and this script file, and then I call my transformer, and after calling my transformer, you can see that 427 of that 1000 rows have intersection with these regions that I defined here, and you can look at those by just displaying the data frame. So that wraps up my presentation here. Again, make sure you check our GLOW website and look at the documentation, join our Slack channel, or contribute to our code base, because this is an open project, projectglow.io. Thank you very much. Back to you, Danny.
Thanks very much, Kevesh. Yeah, if you can stop your presentation and I'll present mine. So thanks very much to everybody who's asked all these great questions. I did want to show my screen just a little bit. Let's go ahead and do that. So some of you was wondering about where you're going to see the notebooks. And so for the notebooks, actually, we have a Databricks Tech Talk location, github.com/Databricks/Tech Talks. So we're going to put the notebooks up there, so that way you can go ahead and take a look at it. We're also do note the fact that this session is actually on our Databricks YouTube channel. So right now it's actually being filmed directly there. So because it is, we're going to go ahead and make sure to put up the link to the notebooks there as well. And also, if you want to continue following up with the conversation, because we're going to prop the video up there on YouTube, by all means, go ahead and engage with us there. And finally, I did want to call out, because this is the Data and AI Online Meetup, we do have some exciting other sessions that are happening, including this one, Encoding Multi-Layered Vega-like COVID-19 Geodata Visualizations. This is from our friends at the Seattle Children's Research Institute, where they're working with COVID-19 data. And what's really cool is they're also using MLflow as a visualization library, in addition to everything else that they're doing. So pretty cool stuff. So again, that's it for today's session. I apologize for running a little bit over time. Kaivesh, Amir, thank you very much for presenting today's session.
Sure.
I'll leave them with the last set of words to say bye, but otherwise than that, I hope to see you guys next week. Amir, Kaivesh, anything you'd like to add before we go?
I just want to mention that the notebooks that we showed here, or even more extensive versions of them, are also available on our on Project Glow website, documentation website, which you can easily go to from projectglow.io.
Yeah,
perfect.
On my end, I'm glad that you guys made it and joined, and I encourage you also to, if you wish, join our Slack channel and the Google groups, and don't be shy, chime in, create your PR, and contribute.
Yeah, great.
Awesome.
Thanks, Danny. Thanks,
awesome.
Henry.
All right, awesome, guys. Thanks very much, everybody. We will see you next week. Take care, everybody.
Okay, bye.